Welcome back to MLT 240. This is part 2 of the Cestode lecture. The next species of cestode we're going to be discussing is Diphyllobothrium latum. This is also known as the broadfish tapeworm. It can last or live up to 25 years. The transmission is through the Pleurocercoid larva in freshwater fish, and it then is ingested by humans. Major symptoms include intestinal obstruction, abdominal pain, vague digestive systems, weight loss, and weakness. This parasite is found in about 1% of the population, usually in people of Scandinavian descent. It can cause anemia and eventually nervous system disruptions due to a B12 deficiency. Because the tapeworm can utilize up to 100% of the host dietary B12. Here is the life cycle of Diphyllobothrium latum. I want to point out and start over here. The cercoid larva is eaten by a crustacean, and then that crustacean is eaten by fish or a fish, and then we humans are able to eat the fish, and that's usually how we get it. Humans ingest raw or undercooked fish. As far as the life cycle, the immature eggs are passed in the feces, and the eggs can mature in approximately 18 to 20 days. Freshwater crustaceans or a copepod ingest the corcidium. Remember, the corcidium is just another name for the oncosphere or embryo. The corcidium then can develop into the larval stage, and the freshwater fish eats the copepod. Inside the fish, the larvae is released and are able to migrate into the fish's muscle, where it's able to develop into the pleurocercoid. That's the infective stage for humans, and that's the point where the humans ingest the raw or undercooked fish. After ingestion of the fish in the humans, the parasite can develop into immature adults, and then into mature adult tapeworms, which reside in the small intestine. The adults attach to the intestinal mucosa by means of two bilateral grooves called a bothria. These are similar to the suckers on the tinea, but they're located on the scolex, and we'll see an example of what these look like in an image coming up. For lab ID of Diphyllobothrium latum, for the microscopic identification, we look for operculated eggs in stool. Operculated means they look like they have a little lid on them, and we'll see that on the next slide. Eggs are usually numerous and can be demonstrated with concentration techniques, so with our flotation or sedimentation. In this particular species, we can see evacuated proglottid, usually in chains ranging from a few inches to several feet. So a lot of times in our tapeworm species, you'll see just one section at a time. It almost looks like a little grain of rice in our stool samples. But this particular species, you can have several sections that are released together at a time. The scolex, or the head, is also diagnostic of this species, but they're rarely released or evacuated in the feces intact. So as promised, here is what the egg looks like for Diphyllobothrium latum. Here is the operculated egg, meaning it looks like it has a little lid on it. And also something to note, it has a terminal knob. So on the bottom side of the egg, there's this little tiny knob. So this is the egg for Diphyllobothrium latum. This next image is showing the actual tapeworm for Diphyllobothrium latum. You see the scolex, or the head. You see the two grooves, or the bothria. So here's a groove, and here's a groove. And again, those help the tapeworm attach to the mucosa. This would be the neck. And of course, the proglottids that are closer up to the neck would be more immature. And just imagine this is a long tapeworm, and this is folded back. These look like more immature proglottids. We can't see the tail end of this worm where the more mature proglottids would be, that would be evacuated or would fall off. Some additional notes for Diphyllobothrium latum. The distribution is temperate regions where freshwater fish are eaten raw or undercooked. Some areas include the Great Lakes of the U.S., Alaska, Chile, Argentina, Central Africa, and some parts of Asia. In Europe, it estimates the infection includes about 20% of Finnish people and up to about 100% of people or residents of the Baltic region. Adults can reach more than 10 meters in length, so that's pretty long. Remember, we're talking meters here, with more than 3,000 proglottids or 3,000 sections. Immature eggs are discharged from the proglottids, and that can be up to 1,000,000 eggs per day per worm, and these are passed in the feces. You can see eggs appear or start to appear in the feces within 5 to 6 weeks after infection. Our next species of tapeworm is Echinococcus granulosus. This is also known as the hyatid tapeworm or the dog tapeworm. It's also known to cause a hyatid cyst. The transmission is that humans accidentally ingest eggs with close contact of an infected dog. Usually the intermediate host is a sheep. The cyst can be found in the liver or lungs. Symptoms vary depending on the location and the size of the cyst. In the liver, which is more common, there's no symptoms until the cyst gets large, and this usually takes about a year after infection. And then there's jaundice and hypertension and/or hypertension. And in the lungs, there's no symptoms until the cyst becomes large. Then symptoms can include coughing and shortness of breath and chest pain. When cysts grow, they can rupture and lead to death, or anaphylactic shock can occur from the cyst rupture. Findings include eosinophilia, urticaria, which is like a rash or hives, or bronchospasm, which is where muscles tighten and cause the airways to constrict or narrow. Here is our life cycle for Echinococcus granulosus. Again, our definitive host is the dog, but an intermediate host could be sheep. It also could include goats, swine, but usually it's sheep. Humans usually become infected when we accidentally ingest the eggs from feces. The adults of the parasite Echinococcus usually reside in the small intestine of the definitive host, which is usually dogs. Eggs are then passed in the feces. After ingestion by a suitable intermediate host, the eggs hatch in the small intestine. The embryo of the oncosphere penetrates the intestinal wall and migrates through the circulatory system into various organs, especially like the lungs and the liver. The oncosphere can develop into a cyst that enlarges gradually, producing protoscoluses and daughter cells that end up filling the cyst interior, and we'll see what this looks like in an image in just a minute. The definitive host becomes infected by ingesting the cyst-containing organs of the infected immediate host. So an example of that would be if the dog consumes the lung meat or the liver meat that is infected from a sheep, then that dog becomes infected. Echinococcus granulosus, as far as lab ID and distribution, we can do serological tests, and we can do the indirect hemagglutination test in ELISA. We can also do a biopsy to look for the hyatid cyst fluid and to try to detect the scolex, the bronchocapsules, or the hyatid sand or daughter cells, but this is usually not recommended because the leakage of fluid from these cysts can cause anaphylaxis. We can also do x-rays, ultrasound, CT scans for detection of cysts or masses in the organs. Echinococcus granulosus occurs worldwide, and it's more prevalent or primary in sheep raising areas where domestic dogs are used for herding. Here's the progression of Echinococcus granulosus. Let's start here with the adult worm. The adult worms, of course, can produce eggs from the mature proglottid and the uterine branches. So here's our egg, and our oncosphere would be the embryo inside the egg, And once that's ingested, of course, you can end up with your cyst inside the tissue of your host. This is what our egg would look like under the microscope for Echinococcus granulosus. You have that thick outer shell. You might be able to see hooks within the embryo of the egg. Very similar looking to our other tapeworm eggs that we've looked at. Here's what the actual worm looks like. The scolex of the Echinococcus granulosus does have a armed rostellum. And of course, as you progress from the scolex down to mature proglottids, those are what's going to fall off in the fecal samples or the stool samples to continue the life cycle and spread your eggs. For lab ID, you can also look for the hyatid cyst in tissues, especially like the lung tissues, and this is what the cyst will look like. And within that hyatid cyst, you're going to start to see the formation of another worm. You're going to have these daughter cells. You're going to have a little scolex start to form. And let's look at the next slide so you can actually see what a liver biopsy looks like and see what a hyatid cyst looks like in tissue. Okay, so this is the hyatid cyst in lung tissue, but again, you have to be careful when you're doing a biopsy of these, because if you cut into these, you could cause a patient to go into anaphylactic shock and release the contents of these hyatid cysts. The last tapeworm that we're going to cover is Diphyllidium canum. This is our double pore tapeworm, or the cucumber tapeworm. The transmission is ingestion of the cysticercoid contaminated flea, or in close contact between children usually and infected pets. This parasite usually infects organisms inflicted with fleas, including dogs, cats, and sometimes human pet owners, but especially children that we just mentioned. Pets may exhibit behavior to relieve anal pruritus, which is itching, and they may scrape their anal region across the grass or carpeting. In humans, there's mild intestinal disturbances. The most striking feature amongst animals and children is the passage of the proglottids, or the sections. These sections or proglottids are motile when freshly passed, so they're mistaken for maggots or fly larvae a lot of times. So here is the life cycle for D. canum. So really, the cysticercoid is in the flea, and the flea is ingested by the animals that is inflicted with the fleas, usually cats or dogs. But if you're a pet owner and you have cats or dogs, sometimes inadvertently we can ingest those fleas as well, and that's how we as humans become infected. The gravid proglottids are able to pass intact in feces, or they emerge from the perianal region of the host. They release typical egg packets, or on rare occasion the proglottids rupture and the egg packets are seen in stool samples. And the egg packets just means there's several eggs enclosed within a packet, and we'll see an example of that coming up. But ingestion of the egg by the intermediate host, the dog or the cat, develops into the cysticercoid larva. The invertebrate host becomes infected by ingesting the adult flea contaminated with the cysticercoid. Dogs are the principal definitive host, but other potential hosts include cats, foxes, and humans. And it's usually children, because children are more likely to come in contact with fecal contaminants because they like to put their hands in their mouth. As far as lab ID for D. canum, typical proglottids or egg packets are found in the stool or in the environment. The different sections of proglottids resemble a pumpkin seed or a grain of rice, especially when dried out. Proglottid or segments have two genital pores, hence the name double-pored tapeworm, and I'll point those out here in an image in just a minute. The adult tapeworms can get up to 60 centimeters in length and 3 millimeters in width. They reside in the small intestine of the host, where they attach with the scolex. Here's a couple images demonstrating decanum as far as the egg. Here's an individual egg. Still you have that kind of thick outer shell that you expect to see in a tapeworm egg. You can see the hooks, but if you jump over here, this is an egg packet. You have lots of those individual tapeworm eggs kind of packaged up in an envelope together, and a lot of times you'll see this in this particular species. Here is the scolex, or the head of decanum. You can see the suckers and the rostellum. You can see that this rostellum is unarmed. Here is an individual proglottid of decanum. And I want to point out these are the genital pores. Remember, it's referred to as a double pore tapeworm. So this is what it's talking about. Each segment or proglottid has a pore on each side. So there's a pore and there's a pore. Double pore tapeworm. If you look over here, this is showing what the proglottid can look like in the feces when it's passed and that proglottid kind of dries out. It can look like a seed or a grain of rice. This is the mature adult worm. Again, the scolex would be down here on this end. You cannot hardly see the scolex of the head in this particular image, but these proglottids or sections on this end would be immature, and as you go on down to the end, the posterior end of the worm, these would be mature, and the ones that were coming off or being released into the feces or stool, these would be mature, and they would have eggs in them to continue the life cycle of decanum, just like any of our other species of tapeworms that we've talked about. This concludes part 2 of our cestode lecture. If you have any questions, don't hesitate to reach out. I'm available during my office hours or through email or after or before lab time.